We are at the Royal Museum for Central Africa, which is based in Tervuren, Belgium, at the outskirts of Brussels. The building itself has been put up by the former King Leopold II when the Democratic Republic of Congo was a Belgian colony, or actually the private property of the king at that time. And he wanted to have a building that was devoted to the activities in the Congo as well as stimulate scientific research in that region. I'm a researcher, I'm an entomologist. I'm specialized in the study of insects, in particular of flies. And my involvement is mainly on what we call taxonomy, which is the identification, naming, giving names to specimens, to flies, so that people know what exactly they are studying.
So I'm Pablo Deschepper, one of the geneticists working here at the RMCA, so that's the Africa Museum. And now we're here in the molecular lab. And here's where we do all the DNA extractions. So when we receive flies, most of the time they're just tissue or the whole fly. And so we do the extractions here, and then afterwards we can use the extracts to have them sequenced. And then we're trying to find patterns in the genetic structure and see if there's like a correlation between certain urban features and the gene flow between populations.
Our involvement in the REACT project is manifold. One of them is first of all the identification. If you want to study an organism, you have to know exactly what the species is, what is the identity of the species. That is where we come in. Secondly, the REACT project is looking at insects that pose a danger to the European horticulture, but their origin is in tropical areas, including Africa. And so because of our linkage with research in Africa, we can provide information on the origin and on the areas where the species originally occurs. The advantage of a museum is that indeed we have a lot of records from the past. And knowing the past and knowing the origin and the history of these invasive species is important to understand what their potential danger would be. You need to know where they come from, what kind of fruits, for example, they would infest. So you need to know a lot of background information which you have to look for in Africa.
So in the REACT project, partners are collecting flies, so they're trapping flies of different locations for two different species, so that's Bactrocera zonata and Bactrocera dorsalis. And they're trapping flies in an urban environment. We try to figure out if there's like certain features within the environment that affect the connectivity between populations of these pest species. And so it's very essential that we have this information from the people in the field where they collected the fly, if there's like a very busy road or if there's like a fruit market in the neighborhood. And then we can really integrate this information into our analyses and interpretation of the results. Prior to the REACT project, we had another project, the fruit fly IPM project, and there, for instance, for Bactrocera dorsalis, we looked at invasion pathways of the species into the Indian Ocean islands. So you have Madagascar, the Mascarenes, Mauritius, Reunion Islands, and there we saw that contrary to what you might think, there's not one invasion pathway coming from Africa, but there's in fact two invasion pathways like colonizing these islands from different directions. If you're targeting one trade route, there's still the opportunity of the species coming in from another direction. So you should invest, like the people making policies, should invest in strategies that tackle the different routes of invasion and not focus on like the most convenient solution for the fly to get into a country.